Okay, we need to get started. We've got another great group of speakers coming up. And our first speaker, our invited speaker, is Dr. Hutton Kearney. And Hutton obtained her doctorate in genetics and molecular biology at the University of North Carolina at Chapel Hill. And she completed postdoctoral training and a fellowship in clinical cytogenetics also at UNC, followed by a fellowship in molecular genetics at Duke University. She's currently the director of the cytogenetics laboratory and associate director of the molecular genetics laboratory here in Asheville at the Fullerton Genetics Center. And some of the other hats that Hutton wears in her spare time include chairing the FACMG Laboratory Quality Assurance Committee, being the president of the American Cytogenetics Conference, and serving as editor of Genetics in Medicine. We're really pleased that Hutton is going to give us some advice today on navigating uncertainty and unexpected findings in whole genome array testing, a discussion of interpretation and reporting strategies. Thanks, Hutton.
I should warn you, I'm not going to give a lot of advice. It's more of a discussion of anxiety. Got it? Okay. The second warning is I don't do formal presentations very well. I do better with discussions. So I would rather, if you have a question, you just interrupt me. It's a very small room, and waiting 45 minutes to ask the question that may no longer be relevant. Or I can repeat it if it's, however you want to do it. So do feel free to keep this informal unless the taping prohibits that. So I'd just like to start off with what I had when I began my fellowship, and I feel still pretty young in the field, and I'm still amazed at how quickly technology has changed our field. And the basic tools that I had when I started, there was the karyotype. If you found an abnormality, if you were lucky enough to see it under the microscope, you had very little resources to check to look for what might be the phenotypic outcome. And this was our best tool, this wonderful catalog of imbalances. And we had FISH to determine if it was there or not, but just so very little characterization of the gene content of the cytogenetic aberrations. But one nice thing about it is that by the time you could see it, you could almost guarantee it was pathogenic. There was very little uncertainty as to whether this was going to have a phenotypic effect. There are, of course, violations to this, but by and large, if you saw it, you could talk to the clinician about a patient outcome. Leap forward about, 10 years or less. The American College is now on board. This was a paper in 2011 where we said, game on, we're ready for whole genome analysis. Please quit trying. This is really geared to manufacturers and FDA. Quit trying to make syndrome-only arrays. We're capable, we're educated, and we do not want to dumb down our products. We want to look at the whole genome, and we will do the interpretation even if it's hard. So be careful what you wish for, because we're now dying, I would say. I'm exhausted. I love it. I think we're all pretty tired, but I think it's a worthwhile pursuit. And so I'm just going to discuss the trials and tribulations of the kind of information that we find with these whole genome technologies. And if any of you do Pinterest, this is Anxiety Girl, which is kind of my superhero. So my old anxiety used to be, have I looked at the karyotype long enough? Did I find the aberration? Were the chromosomes long enough? Now, I know whether the aberration is present or not, and I generally know to the exon level what genes are present. So that piece of ambiguity has been fixed. But now, my new anxiety is, have I looked at enough resources? Did I type in this gene to enough databases, and have I touched enough information to provide an informed and adequate interpretation? Or did I miss something that my colleague would have found if they had this case? And so most of my talk will be about that. So what do you find if you're interrogating the copy number? And this obviously applies to sequencing too, but I'm going to limit this to arrays. If you're looking at the copy number of nearly every gene in the genome, what do you think you're going to find? Well, you're certainly going to find pathogenic abnormalities, but you find a whole lot that you don't understand. And a lot of people like to call these VOUSs, but I think Bill coined the term more adequately to reflect our frustration. We like to call them findings of uncertain significance, which has a beautiful acronym. And typically we think of these just with this, CNVs where we don't understand the clinical significance. But I'd like to expand this definition and through the talk about this, but also talk about CNVs that we understand, but we need another piece of evidence in the patient. And so it's uncertain because you don't know the context yet as to whether it's pathogenic. And homozygosity. When you see homozygosity, it's uncertain until you have other pieces of evidence. So I'll talk about all three. So most labs try to strike a balance in what they report. So when you look at the entire genome, you're going to find information that we don't understand or that we weren't expecting. So what should be in the clinical report? Should we only include those findings of known clinical relevance or those of known clinical relevance that the clinician was asking for? Or should we include all technically valid findings? And I would say most labs find themselves somewhere along this continuum, some closer to the one side than others. And clinicians are the consumers of these laboratories, and they pick the lab that has a reporting style closest to what they're looking for in their patient population. The American College wrote guidelines for interpretation of constitutional copy number variations, and I will say I learned more working on this document than in both of my fellowships combined. It was a great experience, but mostly I learned what I don't know, and I think I'm going to share a lot of that with you. We did recommend categories of clinical significance. Some people think that we have 5 categories of clinical significance, but I'd like to stress that there are really 3. There are those findings that we deem to be pathogenic, that we understand and we know have consequence in human disease, those that we do not understand, that we're uncertain about, and those that are benign. And certainly you can further subdivide your uncertain category into likely benign or likely pathogenic. But I feel strongly, and the writers of the document felt strongly, that the most important thing to communicate was that we didn't know. Because you can take your best guess all day long, but if you're uncertain, that's the most important thing to let your physician know. And just the other day, this was about a week ago, on one of the LinkedIn listservs or boards that I was looking at, I found this quote, and I won't say who wrote it, but I think I took exception to it. So it was, with CMA testing, some labs worked harder. And I think it's the harder part that bothers me, and tried to label more variants as likely pathogenic or likely benign, and that the lab has a duty to report as few uncertain findings as possible. And certainly we have a duty to work as hard as we can and to give as much information to the clinician that ordered the report as possible, but I don't feel that it's necessary to take a guess in my report. I will use likely benign and I will use likely pathogenic, but I use it very conservatively because I've been wrong a lot. I used to use it more. And the truth is, we often do not know, and it's time to stop apologizing for that. As long as we've done an exhaustive search, I think the entire community needs to understand that a lot of what we're seeing now with this whole genome exploration is uncertain. And if we can start getting used to that, we can start having more clearly communicated reports. This is a little bit soapboxy, so sorry. So I'm going to go through our process for a systematic evaluation and clinical interpretation and just show you some things. I certainly can't cover all of it, but the first step we recommend is a familiarization with the well-established contiguous gene syndromes, the known microdeletions and duplications. And this is important because if you find a CNV in the genome and you go to OMIM or PubMed and you look at every gene, many times you're not going to identify the fact that this is part of a contiguous gene syndrome, especially if the critical gene has not been elucidated yet. So it's very important that you have some understanding that this is the TAR syndrome region, this is the DeGeorge syndrome region, so that you can cite the appropriate literature. And PubMed, it's a great resource, but sometimes you don't know what keyword to type in to return the right publications, and sometimes typing in the band is helpful. But I really would love to see MESH subheadings that were genomic coordinate based. It would be a lot easier to find the relevant literature if we would work in coordinate space in the literature. There are resources to help you understand the well-established syndromes, certainly OMIM, Gene Reviews, Decipher, and the literature itself. I just want to point you to one really helpful resource. Decipher's done a great job. It's not comprehensive. There's still some contiguous gene syndromes that are not on this list, but they have a pretty good list with a clinical synopsis and the coordinates of the different recurrent syndromes. And if any of you use bed files or want, we have been collecting hosts and hosts of bed files of these types of things. If you ever want some of these resources, just email me and I'll provide that at the end of the talk. The second thing would be consideration of size. And we had a very, very short, maybe a couple of sentences about this. Certainly size has a correlation with likelihood of pathogenicity, because the bigger the CNV, the more genes, and the more genes, the more likely that you've tapped into one that has copy number sensitivity. But a single base pair in the genome can be lethal when deleted, and a 4 to 10 megabase duplication can be completely benign. So size should be taken with a grain of salt, and you should certainly be more interested in what genes are present. Most laboratories do have a size cutoff so that they can clearly communicate with the physician as to what they promise to report, but that should really be the only, just the technical limitations of your platform and your communication to the ordering physician for expectations. That should really be the only reason size is used, in our opinion. Consideration of the genomic content of the CNV is by and large the largest and most important element of the interpretation. This can be done with a variety of resources. I've only listed a couple. If this gene or region is present in gene reviews, count yourself lucky, because it's undergone a really nice clinical synopsis, and you usually have really good resources and genetic counseling notes to aid in writing that report and communicating to the family. Often that's not the case. Most often you don't have such a nice resource, and we can rely on OMIM, if not OMIM, PubMed, or some of the more research-based, gene-based entities, and HGMD. There's many, 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 many resources that you can touch, and the biggest question is how many should you touch, and how long should you spend on each gene and each CNV, because you could spend weeks on one patient, and that's not financially viable, and it's not good patient care. The turnaround time that would result is not appropriate. Just want to stress, and mainly this is for the genetic counselors and physicians in the audience that might not know a lab person personally that explains this process, just to stress how much time it takes, and to be patient with us, and to understand that this is a very, very laborious process. And it takes good scientific judgment. There's just not a single resource that you can go to. Despite everyone's best effort to put together these clinical grade databases, none of them are without error. And I think there was a talk yesterday, I think Bruce was mentioning, how many miscitations there are in the literature, and you just have to dodge minefields. A really nice paper that a group of us worked on, I shouldn't say it's nice if I've worked on it, but Aaron Riggs was the primary author. We went through and outlined a systematic evidence-based review of copy number variation, or genes for haploinsufficiency and triplosensitivity. And we used this as a framework to start a database. It's a nice clinical grade database, but it also has its limitations, which I'll discuss. But the process involved is the genomic region or gene associated with a clinical phenotype? If yes, and this is really important, a lot of people miss this step, is this phenotype associated with dosage sensitivity? So autosomal dominant gene might be due to gain of function, and you have to be very careful when you're looking at a CNV loss that you're looking at the right mutational mechanism to be relevant for the syndrome in question. We ask how many lines of evidence are there to support its dosage sensitivity, and this is important because there are quite a few case publications out there that are just coincidental observations. So a patient with a particular phenotype has a CNV, and unfortunately this gets published, and it is returned on every PubMed search, including the one that the patient does and the physician does, and it may not in fact be relevant to that syndrome. So it's nice to see multiple lines of evidence. It's nice also to see that the genomic copy number is enriched in clinical populations, because sometimes if you have a phenotype that's pretty vague, like developmental delay, and most of who you're studying has developmental delay, you're going to see cases over and over and over with a certain CNV associated with developmental delay, or autism, or seizures, or some of the other recurrent indications. So it's very, very important, not just to say, have I seen this same phenotype multiple times, but do I have the absence of this CNV in normal control populations? And then is there any compelling evidence to refute its dosage sensitivity? And I'll show you an example of that shortly. So the work that we've done, and we've done over 500 genes at this point, we've started with the most obvious ones, can be found at this site hosted through the NCBI. And if you search any gene, you can come up with a gene page with many, many links to the different resources available, but the most important links are the evidence that we've collected for haploinsufficiency and the evidence for triplosensitivity. And in addition to giving it a ranking based on the paper, the scores that are outlined, we also provide full transparent evidence of how that ranking was designed, so you can check us to make sure sure that you agree. Check the primary resources that were used. If you don't agree, you can report it back to us so that we can update the entry if you find something contradictory to what we found. Or if you find a gene that we've not yet reviewed and you've spent a lot of time working it up for your patient, be so kind as to report it to us so that we can start the review. And we're trying to crowdsource this as much as possible because it takes hours and hours and hours per gene, and none of us can wait on this. We'd like to have the whole genome annotated in this way. But even when you do your best job and you work really hard on one gene, it might be a year before you review it. So it goes out of date very quickly. Just a little note on OMIM, which I think is one of the greatest resources around, so don't get me wrong. I think it's a fantastic place to go for clinical grade information, but it has erroneous information too, and you just have to be careful when you're looking. So one of my favorite examples is the gene DOC8, which is on 9p, and some of you may have had reports of DOC8 deletions in your patients. It's a common finding. There is a report of association with autosomal dominant mental retardation. Two unrelated individuals with cytogenetic aberrations both had mental retardation and developmental disability, fairly nonspecific indication. And so it made its way to an OMIM entry associated with the syndrome. But this is allelic to another syndrome. So DOC8 is also associated with autosomal recessive recurrent infection syndrome. It's been reported over and over and over. We have hundreds of carrier individuals with this mutation, with deletions, so it's not specific mutations. So this evidence would suggest that those first two reports are in error, and it was just a coincidental finding. But it's just very hard when you're glancing through OMIM and you're in a hurry. You have to slow down and look at all the evidence. You can't just stop at the first thing you see. And so that's just a note. The other note, and most of this is beware kind of talk, OMIM and other resources are not always current and almost always not comprehensive. So this was a case I had with a HUE1 duplication. Some of you may be aware of HUE1 duplications and mental retardation. I was not, and my process at the time was to use MapViewer through NCBI, and I still use this. And there was no OMIM link for HUE1, but I had a process in place that if there was no OMIM entry, I would also do a PubMed search. And when I did that, I found a very important publication that suggests that duplication of this is associated with this mental retardation in males. I went back and I checked OMIM. It wasn't there. It's there now. I couldn't pull a screenshot today because they've now updated it, which brings me to another question. How much documentation do we need to put in our lab records of the interrogation that we've done? It's nearly impossible to save a paper record or a screenshot record of every resource that you touched, and I can't reproduce OMIM from 2008 when I wrote this report. So we're going to be asked why we interpreted it as such, or why we failed to report the C&V, or why, and we need to make sure that we've collected adequate documentation to defend us on the stand, I suppose. So just a closing note on that, that just to remember that most analysis software, almost all of them, they import links from other sites, so they'll bring in OMIM, or they'll bring in Decipher, or SNPDB. All of them, almost, are static imports. They're taken a snapshot of that database at a certain period of time, and they're not updated except on regular intervals. And so it's important to know what the gap is. So if you're using a tool, how long ago did they look at OMIM, and should I check OMIM? If this was updated a month ago, it's unlikely. But in a perfect world, you're doing real-time searches with all of the resources, because literally daily there are new gene associations with disease published, and you want to be sure that you've tried to at least find all of them before you throw your hands up and say there's nothing known. The last, and this kind of segues into this, the comparison to both internal databases that you keep and external databases. So the database of genomic variants we're probably all familiar with. It's a fantastic resource, but it has limitations as well. And I just want to remind everyone that this is a research tool. Less than 10% of the CNVs in this database have been confirmed, and especially from the early work, the back arrays and some of the low density arrays, a lot of those CNVs are artifact, and they've not been culled from the database. It doesn't have to be culled. The way they're using this is for very large scale research efforts. But just because you see one deletion in a normal control population, one, it might be an artifact. Two, it might be sized incorrectly based on the platform used. Three, you have to go to the primary piece of literature to understand how that normal control population was selected. If your patient has attention deficit disorder and dyslexia, and the control population in the publication that found its way to this database was a heart disease population, they may have the phenotype that you're looking at. So you just have to use it carefully. And this is kind of a way to some labs will take the DGV and automatically subtract anything that was found in the DGV and not look at anything clinically if it's already been found in a normal control population, and I would strongly discourage that practice. There are wonderful, wonderful international data sharing efforts, and these are not all of them. These are just some prominent data sharing efforts that are out there. I would say that Decipher and ICCG are more relevant for copy number aberrations. The Human Variome Project and ClinVar's catching up, but they deal more with sequence variation than with copy number variation. And we intend to participate in these efforts, but I will say that it's difficult for labs to participate in research efforts. IRB is prohibitive. Our time is prohibitive to collect the amount of energy and materials to submit in the way that they need to see this data. And out of frustration and just an acute need, especially in a smaller lab when you don't have a lot of cases under your belt, you need to share experience with other labs. And I, for years, found myself calling my colleagues or emailing my colleagues, have you seen this, have you seen this? And luckily, I have enough friends that are willing to pick up the phone and take the time out of their busy day to look in their internal databases, and I was able to get a lot of information from them. That's not efficient for me, it's not efficient for them. So we needed both our internal database and a way to interact with our colleagues quickly and efficiently and immediately. So ISCA offers this as well, but you have to have, because it's a publicly available research tool, they don't publish which lab reported the CNV, so it takes often a week or more before you can get contact with the lab that reported it if you have more questions, which is not a reasonable amount of time if you're trying to use that information to sign out a case. We also needed a mechanism to collaborate with those larger efforts, and the database we made, and I'm not going to spend much more time on it, but we accomplished all of those goals with this database. I'm really proud of it. It's a super small effort. It's not very expensive. We don't have large amounts of research funding, but it works great. And we have international uptake at this point. One thing I really like about it, because it's password protected, this is not considered a research tool. At least most IRBs that we've queried do not consider this research. This is best practices and case consult. And I know at least for Sarah South at ARUP, who has a lot of cases in this site, and myself, we put our entire report in. And I feel a little bit vulnerable when I do that, because I'm sure some of you would look at my report and judge it. It might be wrong, or you might not like the tone of my report. But I would really appreciate reading yours. I might not use it, I might not use sentences from it, but it's a start, and we can provide the publications that we touched and found to support our interpretation. And I'm committed to continuing doing that, because if you do see an error, and one person actually found a typo in one of my ISCN nomenclature strings, and I was able to amend my report. So it's sort of embarrassing, but it allowed me to correct my report, which should be the most important piece. Just to show you, we have labs from all over the world. Most of them are looky-loos. They just log in and look at our cases. We can't seem to get people to take the effort to put their cases in, but we believe it's coming, and we think it's quite important. Advisory board, many of you in this room actually help us with this effort, and I appreciate it, and I want to just acknowledge the Mission Foundation who supported the project. Okay, so I'm leaving CNVs for a little bit, and I'm going to talk about the other two categories of uncertainty. Really briefly, CNVs that are only pathogenic in context. So a CNV over a recessive gene, if your patient has a phenotype consistent with that recessive disorder, it's possible that this is in fact one of the two mutations that that patient has, but you need more studies. It's still uncertain until you've found the other hit, or it's an imprinted gene and you need to understand whether it was paternally or maternally inherited. But my least favorite, and I was really grateful for the talk yesterday, my very, very least favorite finding are partial duplications. And I'm not talking about intragenic duplications where you can look at how many exons were duplicated and predict frame shift and have a pretty good idea of whether it would be disruptive, but large duplications with an endpoint in a gene. And usually, and usually turns out, 84%? Usually these are not disrupted, and we know that, but we still have the possibility that they are, that they've either created a fusion gene at the interface, or that by virtue of being inverted or some other structural problem have disrupted a copy of the gene. And there are enough scary stories in the literature. This is just one, LIS1, which I refuse to call by its new name because it's too hard to remember or say. It'll forever be LIS1 to me. A disruption of LIS1 from a large duplication that was inverted. So every time we see one of these, and it is frequent, and I know our physicians are sick of seeing this little phrasing that we write. We say something like microarray data can't, I can't even remember my phrasing, but this may or may not be disrupted, and good luck, because we've really not had a tool to follow them. And I think the last surge in Asheville, that was part of our breakout discussion, what can we do? And it looks like there is something to do, but that's an awfully expensive pursuit. So until the costs of whole genome sequencing come down, it's not a reasonable follow-up approach. I'll just share with you 2 cases that we had. These were around 2008 or 2009, where we had a nearly 1 megabase duplication, but the breakpoint ended in BRCA1. And so that was a super unhelpful report. Like, you may or may not be a BRCA1 mutation carrier, and we of course found it in family members in both cases, one from a mom and one from a father. And Jessica Booker at UNC did as much as we could. Sequencing doesn't help in this case, and I can go through the details of that if it's not obvious to you. And we did protein truncation studies, which was the best thing we could do to assess whether we had full-length protein. And we could look at the family history and say there was no family history of breast cancer, but I have to say it was a really unsatisfying follow-up. And I don't know that we did that family any service by saying you might have a BRCA1 mutation, but I don't know how I could not put that in the report. And I guess the question I would ask is when the gene of interest, when you have a mechanism that you don't understand or a small CNV that you can't confirm but you're suspicious of, you tend to be more likely to want to report those if the gene has a high clinical importance, like BRCA1. But the ramifications of being wrong are also higher when you over-report findings in genes that have high clinical importance. And so I would just, it's maybe just a food for thought, like how much responsibility do we have to chase these things down when we see a gene like BRCA1 involved? And I couldn't finish this story for this family, or either family. Homozygosity. This has lately been my favorite topic because it's new. I think I'm exhausted from CNVs, and homozygosity is kind of something new, and I'm starting to be now exhausted by homozygosity as well. We've been using SNP-based arrays since 2007, which you would think would be a nice, healthy dose of experience under our belt, but despite the number of years that we've been looking at these findings, we still aren't sure if our cutoffs are correct or if they're appropriate. And so I tend to talk to my colleagues a lot about this to try to make sure I'm doing it right. And we continue to evaluate our reporting criteria because we don't want to trigger a lot of unnecessary follow-up, and yet we don't want to miss a potential report of clinical significance. So when is homozygosity clinically significant? It might not be. All of us are homozygous at many loci, and we probably have several large stretches of homozygosity. And if you're homozygous for a wild-type allele or a functional allele, all the better. But it may be a hallmark of UPD, uniparental disomy. It might suggest that you're a carrier or that you have a recessive disorder. It might suggest that your parents are related, and often more closely than you would have imagined, and sometimes so closely that it suggests abuse involving the parents of the program that you're studying. And I'll talk about all of these. But the most important thing to remember, especially those of you receiving these reports, and hopefully your lab does a good job of explaining the fact that this is an inference and not a diagnostic observation. If they didn't, please remember it yourself. These are all inferences. There's almost nothing diagnostically you can obtain from looking at homozygosity on a SNP array. It's the beginning of a search for a diagnostic finding. So first you have to understand how much homozygosity is typical, and I'm not going to go through the whole chart, but just to show you that in one study of both inbred and outbred individuals, 3 megabases seem to be the cutoff at which the two populations seem to diverge, where a normal outbred population had very few CNVs greater than 3 megabases. Smaller than that, we all have them. And so we use 3 megabases as a cutoff to filter our results. Let's see. So with this cutoff, we have about 3% of the cases that we report go out with some discussion of homozygosity. And when we launched our SNP array, we thought that UPD was going to be the big bang here. That was why we adopted a SNP array in the first place, is to search for UPD. But what we were not prepared for were how many of our patients had parental consanguinity. We were not prepared to report it, we were not prepared to interpret it, and we were not prepared for the very exciting diagnostic field that came from subsequent autozygosity mapping. And I will talk about this a little bit. Because nearly 1 in 10 cases, whether we report a suspicion for UPD or a suspicion for parental consanguinity, and we say you have this or that block of homozygosity, nearly 1 in 10 times we can find the recessive mutation. That might have gone down a little bit because we haven't had a case in a while, but it's a significant amount of cases. So when do you consider UPD? So when you have an isolated block of homozygosity, and I should have introduced the term, LCSH just means long contiguous stretch of homozygosity. So if you just have one chromosome involved in a big block of homozygosity on that chromosome, you should think about UPD. Doesn't mean it is UPD, but it should certainly get your attention. And I'll just kind of review this for anybody taking boards anytime soon. This is kind of a nice review, but I won't go into it too much. But the typical, not the only mechanism, but the typical mechanism for uniparental disomy is to have a trisomic conception that is subsequently rescued by a mitotic error. So if you have 2 mom 15s, let's call these chromosome 15, then 1 dad, and you throw one of them out during meiosis, if you happen to throw the only dad's 15 out, you're left with 2 15s derived from mom. And typically we draw it like this, and the 15s look like they would be identical. And I'm just going to show you a couple of diagrams that I will not try to teach, but just to show you, it's not like this. It's much more complicated than that because of meiosis. Meiotic recombination generates regions of isodisomy and heterodisomy, and that's important to remember. So this is just a schematic of meiotic recombination, and just a close-up when you have trisomy rescue, and this is a meiosis 1 trisomy rescue, you're going to have regions of heterozygosity and regions of homozygosity, and those 2 15s derived, or whatever chromosome that is, derived from the single parent. And the placement and the size of those varies depending on a very random pattern of recombination. You can't always predict it. Not going to show you this chart, but just to show you there are lots of different ways you can generate UPD and lots of different expectations for the recombination patterns and the position of homozygosity. And if you're a lab interpreting these arrays, I would just recommend that you review it, just to be aware in general of where you would expect the homozygosity to be. And the point I want to make to the whole room is that if you have a meiosis 1 non-disjunction, which is the most common, especially in females, error, and you don't have any crossing over, which you might argue would increase your chance for a meiosis 1 non-disjunction event if you don't have the crossover, you're going to generate 2 maternal chromosomes that are completely heterozygous, no homozygosity at all, and you would never pick this up by surveilling for homozygosity. And so it's super, super important that the clinicians ordering the test understand that this is not a diagnostic assay for uniparental disomy. If you're suspicious of UPD, you should order a molecular test for that suspicion. We find it serendipitously, but it's not a diagnostic test. We also see very, very small regions, so we might miss it because the region is just too small. This is not the smallest one I've seen, but it's the only one I have a slide of. This was a 5 megabase terminal in a Prader-Willi. Sarah South has had a case of 3, which barely makes my cutoff, so I'm not sure if we had a 2 that I would see it. I hope that I would see it visually, but I'm going to miss some of them if they're very small. I'm also going to miss them if the UPD occurs in a background of consanguinity. So if there are blocks of homozygosity throughout the genome because the parents are related, a block of UPD that's a block of homozygosity that's UPD related is going to get lost. Another reason not to promise this is a diagnostic assay for uniparental disomy. And these are just tips. I'm just trying to teach some things because this is new to some people. Also remember that when you're thinking about whole chromosome uniparental disomy, where the homozygosity is, a lot of people want to see it over the Prader-Willi-Angelman region because that's the imprinted region, but it doesn't matter where it is. It's a secondary hallmark of uniparental disomy. What matters is that both of those chromosomes went through the maternal germline and were imprinted in a pattern for maternal expression. The placement of the homozygosity is just a byproduct of recombination. That isn't true if you're talking about segmental UPD, which is more rare, but is certainly something that we see in Beckwith-Wiedemann. If it happens somatically, those chromosomes didn't go through the germline, and the only region that's uniparentally derived are the regions that are homozygous. just going to say it as a note and move on because that's not really the focus of the talk. Okay, that's UPD. Now I'm going to talk about consanguinity. This has been probably the most difficult, more difficult than incidental findings, at least emotionally for us. It's been a really, really tough kind of case to deal with, and I feel very fortunate to work in the lab, and when I say deal with it, I really just pass it along to the clinical team to deal with it. And unfortunately, Carolyn Wilson, our genetic counselor, has seemed to get the lion's share of these cases, so she's now like the world's expert in first-degree parental consanguinity. But when you see 25% of the genome homozygous, or 30, it varies, you can be pretty suspicious that those parents are very, very closely related, probably first degree, brother, sister, father, daughter being the most common. And unfortunately, we see this a lot more than we expected. We saw it the first week we went live, and I'll show you that case. You can, from the expectation for the coefficient of inbreeding, which we all studied for our boards, you can make a pretty clear prediction if your population is outbred as to how closely the parents are related. We make this calculation just as an FYI, but we don't report it. We don't report our suspicion. It's kind of nice when we get a case like this where we get 7.4% homozygosity and we predict, they're probably first cousin, and the clinical team says, yeah, they're first cousins. It makes you realize that you're thereabouts estimating it correctly, but it's not really relevant. Who cares? It's not our business how closely the parents are related, unless we feel that the mother, or more rarely the father, has been abused. The reason why we can't make this calculation with any confidence is that meiosis is random. How many crossovers, where they're placed, generates different sizes of homozygosity. There can be multiple loops. You can have double first cousins or a population that is in a very small gene pool and amplifying the percent homozygosity, even though the parents themselves are not that very related. You can have adoptions and non-paternity, which complicate your assessment, suppressed recombination. And I'll just tell you one case, just more of a scare tactic. This was a 17-year-old child, and he had some clinical features, which aren't really the point of the talk. We had 18% homozygosity, so we weren't sure if it was a first degree or second degree, but we knew it was pretty close in the mother. And when the clinical team interviewed the mom and asked are you possibly related to the father of your child, she said, I'm adopted. I have no knowledge of my biological family at all. My husband has a very large family, lots of half-siblings. He was raised in foster care. He has a very clear understanding of his family lineage, but I don't because I was adopted. So we suspect strongly that the mother was adopted into the same community and perhaps married. She's happily married to this man, married her half-brother or her full brother without knowing that they were biologically related. And this has implications for sperm donation as well. So unrestricted sperm donation in the same community, people are going to find their sibs or half-sibs unknowingly. The mom had a hysterectomy. The child was, it was not a situation of abuse, and we didn't end up talking to that family about their relationship. But it was the first week we went live. We had no policies in place. It was devastating. We were not ready for this kind of report. So if you haven't launched a SNP array, please prepare yourself for these cases. I'm going to just move forward just to let you know that there are guidelines now from the American College of Medical Genetics about how to deal with suspicion for abuse and how to report consanguinity. They're available now at the ACMG website. And I'm going to skip I'm just going to talk to you about why I bother. So if it's not UPD, and we don't think it's abuse, why would you even report consanguinity in the parents? And the reason is we're very successful in finding recessive disease, especially when you have a close relationship with your clinical team. And when I say autozygosity mapping, most of us think of it as the research tool to identify a new gene in a consanguineous family related to a disorder, and it's been a super useful tool for gene discovery, but that's not really what I'm talking about in a clinical situation. It's not feasible for us to do gene discovery on the basis of one, usually small family. But what we can do is if the clinicians have a very specific indication, like Usher syndrome, we can tell them which of the Usher syndrome genes are in homozygous intervals, which can help narrow down the subsequent testing. If we suspect a rare disease for which there's a very expensive test or no test available, and we're considering developing such a test, it can increase our likelihood of success and our confidence to invest that amount of money, either the patient's money or our money, to follow that finding. It's been very, and just let you know that there's a tool, and this is a tremendously long URL, but if you just Google ROH tool, it's a really nice tool out of the University of Miami to help you with autozygosity mapping. And I'm not going to go through the features of it, but just to let you know it exists. And so I'm going to skip this for time because I'd like to get to questions. This is just our strategy for homozygosity reporting. So rather than concluding statements, I'm just going to leave you with questions because I often find that I have more questions than answers. One of them that I struggle with, when has our lab done enough to characterize an uncertain finding? And I'm almost never sure I'm finished, and that is the honest truth. But I guess working with enough other labs and working with groups of people doing the same thing, I feel like I'm doing at least as much as my colleagues, and I'm trying very hard, and I think I have to sleep well. My turnaround times, again, are abysmal, which I'm not sure is very good either. A question that I've not answered, is this research? Once, when you have an uncertain finding and you do other tests to characterize it. So let's say I say you have a suspicion for UPD, and then that family orders a UPD test and pays for it, and it's not UPD. What if my reporting criteria were wrong? Like, I feel a great responsibility to try not to trigger unnecessary testing. And when we're starting to study family members, it starts to feel like research, and insurance starts to not want to cover this. And a lot of labs just do it for free, but we were hemorrhaging. We can't do it all for free. So who's going to pay for the follow-up? I asked this question earlier, how much documentation do you need to keep for your evidence that you use, either to not report a finding or to report it? And the last question that I'll leave you with is our community's intolerance for uncertainty forcing us into inappropriate or inaccurate diagnosis? And I would argue that the answer is yes, and just try to encourage people to communicate their uncertainty confidently, because it's not knowable in many cases. I'd like to acknowledge the Fullerton Genetics Center, specifically my array team, who are the best group of technologists in the land, and my very numerous, very smart, and generous with their time and cases colleagues that have helped me in innumerable times. And hope you've enjoyed Asheville. And I don't know that I have time for questions, but yeah. Oh, I'm clapping for myself. How funny. Questions or discussion?
It was a really great review, very interesting. I'm going to ask you the hard question. The family in whom you found significant evidence of consanguinity but chose not to tell them about it, what about their risks for future kids?
In that case, the mom had a hysterectomy, and that was a big factor in our decision not to report it. If there was potential for more children, we would have talked to them about it.
Great, thanks.
So, to use your example of if you had a patient where a clinician thought the patient had Usher syndrome, if you had not found excessive levels of AOH in that patient, would you then go back and look at even the smaller regions that were less than 3 megabases apiece and sort through those, see if any of those matched up with any of the Usher genes?
I'm not sure about the less than 3 megabases, but in that case, actually, our trigger is 2%. That's about the percent at which we'll issue a report. It was 1.5. It wouldn't have triggered a report, but because the clinician had let us know it was a recessive indication, we took the time to look at the Usher genes and the entry holes. But I wonder, because there's so much population homozygosity, less than 3 megabases, that it really dilutes your search. And I think if you start looking in those really small regions, you're going to have a lot of fruitless follow-up because they're homozygous in a lot of people.